This video presents a didactic approach to the principle underlying the COVID-19 vaccine. The human body consists of 30,000 billion cells. These make up all the tissues, namely cerebral, muscular, and connective tissue, as well as blood and immune cell lines. The main intracellular components are the plasma membrane, the ribosome, the lysosome, and the endoplasmic reticulum. The plasma membrane is the extremely resistant envelope of the cell. Thanks to its hydrophobic nature, it protects against the cell flooding. It is, however, perforated by pores which allow the exchange of proteins and molecules. The endoplasmic reticulum is a direct extension of the outer nuclear membrane which houses the ribosomes. The ribosome is a veritable protein factory which receives the coded message for the production of proteins. Lysosomes act as the waste bins of cells, capturing and destroying waste, especially messenger RNA, immediately after transcription of its message. The nucleus is the real nuclear power plant of the cell. Impervious to the entry of RNA, it is, however, responsible for its manufacture on demand. The nucleolus is the DNA warehouse. Situated inside the nucleus, it contains chromosomes of linear DNA which constitute all the genetic capital for cell division. Viral attacks work through a lock and key system. The surface protein, known as the spike protein, is the key. The lock is the major histocompatibility complex located on the cell's plasma membrane. Viral infections occur when the two elements combine. This allows the virus to penetrate the cell and multiply in its cytoplasm by making use of its energy. The cell will therefore die by apoptosis. The nucleus, the genome, and the DNA chain. The nucleus also has some free strands of DNA next to the nucleolus. The nucleus is like a library of recipe books. Chromosome DNA and 30,000 genes are the recipes used for producing proteins and molecules. 23 chromosomes come from the father, in red, and 23 chromosomes from the mother, in blue. They originate from cell division at the time of meiosis and mitosis. When a protein is required, the necessary DNA recipe is released in the nucleus to provide the corresponding messenger RNA. To do this, the DNA will combine with a sort of 3D printer, the RNA polymerase enzyme, which, rather like undoing a zip, will split the two strands of the DNA's double helix into two free strands of RNA, in the example, a purple one and a black one. DNA is like a marine anchor chain coiling together 30,000 genes. A base pair measures 0.5 billionth of a meter, a nanometer. There are thus 2 meters of DNA per cell. There are 30,000 billion cells in the body, giving a total length of about 100,000 billion meters of DNA. The cells of the human body thus contain an overall length of DNA corresponding to 2.5 million times the Earth's circumference. From DNA to messenger RNA. On leaving the 3D printer, the messenger RNAs leave the nucleus through a pore in the nuclear membrane and make their way into the cytoplasm to find a ribosome. Like a train moving freely through the cytoplasm, messenger RNA carries the encoded message to the ribosome factory's computer. This message corresponds to a triplet codon for targeting this codon's specific amino acid. This is the process known as translation. The transcription process then sends a transfer RNA to the stores to find one of the 20 amino acids corresponding to the triplet codon. The elongation process will form a molecule by stacking together the various amino acids encoded by the recipe. messenger RNA assigns the transfer RNA for each triplet codon. For example, the AGC triplet codon will target threonine, and UUU will target phenylalanine. In the example, the transfer RNA is represented by the body of the man, and the amino acid is represented by a red, green, or blue head. Each transfer RNA transports its own amino acid to the ribosome factory for attaching and sticking together with the previous and subsequent amino acids. Once the molecule is synthesized, the messenger RNA is immediately destroyed. The resulting waste is collected and then discharged into the lysosomes. This chart illustrates the fundamental difference between the cycle of endogenous cellular RNA and exogenous vaccine messenger RNA. The former is produced in the nucleus and leaves it to go to the ribosome factory and manufacture proteins on demand. The latter, which is prepared and modified in the laboratory, will have no contact with the nucleus or its genetic capital, and thus cannot modify it. COVID-19 and the vaccine. The spike protein, the key to opening the cell. The antibody, the padlock. The virus infects the cell. Intramuscular injection into the deltoid muscle of a vaccine RNA strand combined with a lipid nanoparticle. This ensures its stability and prevents it disappearing too rapidly before contact with the immune cells. The vaccine RNA, having reached the immune cells, enters the cytoplasm and then the ribosome. The ribosome then decodes the message to produce the antibody mold, which will close around the key to the spike protein like a padlock. Padlocked by the antibody mold, the virus no longer has access to the cell lock. No longer able to enter the cell, the virus is repulsed, and having failed to collect the necessary energy, it dies.